أنا الدكتور إسلام الدبابسة، فلسطيني الجنسية، موجودين حاليا في أوكرانيا. راح نتحدث اليوم عن أحد براءات الاختراع اللي تم تسجيلها، وهي في مجال علاج العقم عند الرجال. بداية حبيت أعرفكم على ما هي الخلايا الجذعية أو أنواع الخلايا الجذعية التي يتم استخدامها، خاصة الخلايا الجذعية التي يتم استخدامها في هذا النوع من العلاج. To go with English language or to talk Arabic language, which better for the auditorium? Okay. There is three kind of the stem cells. The first thing, it's embryonic stem cell. Many people, they don't understand still that embryonic stem cell can be taken from adult body. It's as a pluripotent cell. It's a germ cell which can be in the bone marrow, or it can be in the skin, or in the peripheral vessels, peripheral blood. Second one, it's adult stem cell. It's MSC. It's a stem cell which can be taken from blood cell, from the cord blood, from the skin, from the fat, from its typical stem cell, which can be just stimulate or activate when you inject, when you use, can be activate cells in our body. And the third kind of the stem cells, this is endothelium pluripotent cells. In Arabic, it's الخلايا الجذعية المستحثة. This is kind of the stem cell. We can do it with them, manipulation that we can making reverse for them, and we can make genome engineering, which mean we can block knockout, making knockout for some cells or chromosomes, and we can activate another kind of the chromosomes. What we are doing in our clinic, there is stem cell treatment and there is genome reprogramming for the cells. And through this patent or through this lecture will be with you Dr. Amina, that she is my assistant and my daughter, and she is scientist and gynecologist, and she will go through the our presentation and through our patent and our one of the publication what we did.
So, good afternoon, everyone. My name is Dr. Amina El-Dababsa. And today, the topic of this lecture is, as you may know, stem cell-derived spermatogenesis for the management of male infertility. This is a project that was mainly started as an idea by Professor Dr. Islam El-Dababsa, and this is mainly one of the reasons why he has gained such a name of a professor as well. This was one of his biggest works in Ukraine for I.D. Stem Cell and Genome Institute. So big thanks for the scientific breakthrough that he made. So this idea started back in 1999, but was only proven to be fully true by independent committees and patented in 2026, January 2026. Okay, so Professor Islam El-Dababsa, as you may know, he's the director of I.D. Stem Cell and Genome Institute. He's the founder of I.D. Medical Group and the medical director of I.D. Clinic, where all the wonders that have been discovered in the institute take place in clinical trials and clinical application. He's a professor in genetics. He's a stem cell researcher since 1999. He has a PhD in cardiovascular surgery. He's a SSCR and ISCT member as well. And from 2025, I.D. Clinic heading with our team leader, Professor Islam Al-Dababs, as well as me, myself, as the chief scientist of the institute. We are now affiliated with the American Board of Regenerative Medicine, which means that all of our studies that are within the topic of regenerative medicine, within the field of regenerative medicine, are compliant with everything that the American Board of Regenerative Medicine requires. We are now teaching specialists, doctors that would like, even dentists and physiotherapists, and anyone from applied healthcare. We can teach them to gain this subspecialty in regenerative medicine, and help them get the certification. This is just briefly our team in Ay.Di clinic in Kiev, Ukraine. Okay, so let's start with the talk. What are the objectives of today's lecture? I would like to start with why did we start this? Why did we have this idea? Why did Professor Islam ever think of the problem of getting spermatocytes from somewhere else other than where they naturally come from? Because the issue of male infertility is currently growing, and it is going to, or looking like it is going to become one of the leading causes of infertility with couples, because currently it is between 40 to 50% that the male infertility is the cause for infertility in couples. Before that, it was mainly women, because they are the ones who get all of the checkups and the blame for infertility. But now, since science is going deeper and deeper, we are now finding more and more causes why males have the effect on infertility as well. Then we will discuss what male infertility is, what causes male infertility, and what are the available treatments. And then slowly we will go to discussing our patented method. Okay, I'll start with male infertility. By the WHO, it is defined as the inability of a male to make a fertile female pregnant for a minimum of at least 1 year of regular unprotected intercourse. Before that, that was 6 months. Since 2024, it is 1 year. The male is solely responsible for about 20% of cases, and is a contributing factor in another 30% to 40% of all infertility cases. So that is now the statistics from 2025, which is now growing as well. Now, what is painful is that the biggest percentage of male infertility is unfortunately in the MENA region, which is the Middle East and the Northern Africa, and they are well above Central Europe, Western Europe, Southeast Asia, and several other countries among the world, with the biggest leading two are in Turkey and Morocco. Okay, what can be the causes of male infertility? Obviously, we can divide them into acquired or congenital. So among the congenital causes, we have some syndromes like Kalman syndrome, Prader-Willi syndrome, Klinefelter syndrome, Young syndrome, Sertoli-Cell-Only syndrome. From acquired, we would include environmental toxins in there, as well as some medical causes. For example, when there is some sexual dysfunction, premature ejaculation, anejaculation, low sperm count, as well as some cancers within the testicular tissue, chemotherapy, radiotherapy that will cause infertility in the future. And among all of these, there is obviously always idiopathic causes. What are the current available treatments for male infertility? So conventional treatments for male infertility focus only on improving sperm quality. But for the definitive absence of sperm, which is azoospermia, or an extremely low count of sperm, as in oligospermia, there is no current treatment. They are just conventional, so vitamins, hormonal therapy, or some surgery that can help improve the sperm quality. But what would a patient with a diagnosis of azoospermia do? if he does not have sperm count at all, maybe some chromosomal or genetic abnormalities that stop his body from producing any sperm at all, there is no treatment for those people. And doctors tell them they have no hope in having their own child. Among another types of treatments is assisted reproductive technologies with intracytoplasmic sperm injections, IVF. However, again, for patients with total loss of sperm or total absence of sperm, this option is not available. Available only with a donor sperm, which is where we hit an ethical issue with acceptance in the Middle East and the Northern Africa region. So here is where we start thinking about regenerative medicine and its potential in helping such patients. So at first, obviously, stem cells, as their function, as Dr. Islam has explained, stem cells are undifferentiated cells that can develop into any types of tissue. So this is where we start thinking it can help the testicular tissue grow some Sertoli cells, so then the spermatocyte can grow, but that's with the condition that the Sertoli cells are present and the Sertoli cells are capable of producing sperm. Again, we're going back to the same issue. Patients with azoospermia, patients with chronic chromosomal or genetic disorders where they possibly cannot produce any sperm hit the same issue. Not even stem cells can help them produce the sperm. As Dr. Islam explained, types of stem cells and what is stem cell therapy. The main type that we are looking at is mesenchymal stem cells. They can be taken from adult stem cells or an embryonic. So from adult stem cell, they can be taken out of bone marrow, adipose tissue, or even the blood. Now, by their function, they are divided into adult or embryonic. And now embryonic, I don't mean they are taken out of an embryo. I don't mean they're taken out of anything that is surrounding the embryo. But by their function, they are completely undifferentiated, high potential cells. Some people call them new cells. Some people call them germ cells, embryonic cells, pluripotent cells. There is no definitive definition for this type of cells because regenerative medicine grows every day and new definitions come out every year. So what are induced pluripotent stem cells? Another type of stem cells, but these are, like Dr. Islam explained, these are induced. These are genetically modified, lab-generated cells that undergo reprogramming from an adult cell. So they're taken just like an adult mesenchymal stem cells. They're taken out of skin, blood, adipose tissue, bone marrow. After that, they're reprogrammed in a lab to become this pure, undifferentiated, the purest form of cell, which is pluripotent, induced pluripotent. However, here we hit the problem of
recording in progress.
Sorry. Okay, so here we hit the problem of whether this lab generation and lab reprogramming can damage the cell, can damage its genetic value, can damage its information that is inside of the cell. And this is where all the problems arise with oncology, with uncertainty of where the cell is going to go. What is it going to do? How is it going to behave? Still, clinical trials have not been commenced yet, but it is a promising approach. Okay, now we will discuss basics of human spermatogenesis. So if we think about stem cells and we think how can we use stem cells for helping patients with azoospermia, we would think, okay, since a stem cell can be differentiated into absolutely any type of cell, why wouldn't it become a germ cell, which is cell that then becomes a cell with sexual function, so like an oocyte or a spermatocyte? Here we need to start thinking deep. How does a sperm grow out of a stem cell inside of our body, inside of the male testicle? That's what I want to explain now. Here in this picture, you can see the typical anatomy of the testicles and the seminal vesicles where the spermatocytes are produced. So the total duration of spermatogenesis is approximately 64 to 74 days in humans, maximum 75. After that, they can be pushed out from the testicle. The process is divided into three main phases. The spermatocytogenesis, which is the main parts of mitosis. After that, the meiosis, and the differentiation, which is the spermiogenesis. The first phase, spermatocytogenesis. It is primarily a mitotic phase, like I mentioned. It begins with the SSCs, spermatogonial stem cells, located near the basement membrane of the seminiferous tubules and lasts approximately 16 days. This is where the stem cells undergo asymmetrical division to both self-renew and produce differentiating type A spermatogonia. Through successive mitotic divisions, then they give rise to type B spermatogonia. So I'll go back to the picture of the anatomy. So this is the testicle, and this is at the base membrane of the testicle. This is where we have those stem cells, spermatogonial stem cells. So this is where they start proliferating, undergoing mitosis to type A and type B. But let's say we have around 20 to 50 billion stem cells. After filtration, after mitosis, the natural filtration of our body, we have around 40 billion. After the second round of mitosis to type B, we will have around 20 billion. So here I would like you to pay attention to the key markers of such a process inside the body. So there's several key molecular markers that will help you identify the progression of the mitotic phase. So when the cells begin to differentiate, they express KIT or c-KIT, a receptor tyrosine kinase that indicates the commitment to the spermatogenic lineage. So that means that this specific stem cell is going to become a sperm cell sooner or later. And then the STRA8, stimulated by retinoic acid 8, it is another critical marker that is activated by the retinoic acid that initiates entry into pre-meiotic DNA replication and is considered a hallmark of differentiating from the mitotic to the meiotic phase. So two markers, one to identify whether they are in the spermatogenic lineage, and the other one is to identify that they are ready to progress from mitosis to meiosis. After the mitotic expansion, the primary spermatocyte, which is type B now, they enter the meiotic phase, which spans approximately 24 days. Again, it is divided into meiosis 1 and meiosis 2. In meiosis 1, the primary spermatocyte, they undergo recombination and homologous chromosome segregation, resulting in secondary spermatocytes, which are a haploid, very important, haploid in chromosome number, but still contain duplicated chromatids. These secondary spermatocytes, they rapidly proceed through the meiosis 2 stage, where they produce the round spermatids, each containing a haploid set of chromosomes, and now a single chromatid per chromosome. The presence of a gamma H2AX, a phosphorylated histone variant, signals sites of double-strand breaks essential for homologous recombination. So again, this is a marker. The gamma H2AX is again a marker of the presence of meiosis 1 and 2 stage in specifically secondary spermatocytes. Additional markers are DMC1 and RAD51. They are recombinase enzymes that are involved in the repair of these breaks and the alignment of homologous chromosomes. So these two markers, they identify whether the spermatocyte is going to be healthy or not. So these markers are used not just in anything related to spermatogenesis. They are also used to identify healthy chromosomal division. After that, we're going to the final stage, the third phase of spermatogenesis, which is called the spermiogenesis. This is the direct formation of a spermatocyte. It is a post-meiotic transformation process lasting another 24 days. During this process, brown spermatids undergo morphological and structural changes to become fully differentiated, elongated spermatozoa. So this is more not chromosomal or DNA divisions. This is specifically just morphology and the structure of a spermatocyte. During this phase, several key processes take place. These are very important because the structure of the spermatocyte helps it break inside the egg, inside the oocyte. So without a proper structure of a spermatocyte, it is still going to be unable to produce an egg, to produce a baby, an embryo. So acrosome is one of the most important ones because this is derived from the Golgi center, the Golgi vesicles. And after that, we have the elongation of the nucleus with tight chromatin condensation inside the acrosome, which is how we identify it. And also the second most important, second also important part of the spermatocyte is the flagellum. After that is shedding excess cytoplasm as residual bodies. The chromatin remodeling is a central event in spermiogenesis, the third phase. And it is mainly driven by the sequential replacement of histones with TP1 and TP2, and the protamines PRM1 and PRM2. These are also can be used as markers of healthy structural and morphological spermiogenesis. So this allows for the tight packaging of DNA essential for the protection and delivery of the paternal genome inside the oocyte when the embryo is formed. Another very important markers that I would like you to pay attention to are acrosine and SP10. Acrosine, it is an expression protein. It is a protein that is expressed from the acrosome itself, which gives the acrosome color as well. So these are markers of a normal acrosome formation, the acrosine and SP10. The acrosome is important for the oocyte penetration. Without the acrosome, the sperm itself will not be able to break the shell of an oocyte. So when we do the test of the sperm, an analysis, the sperm analysis, and there is sperm, they're mobile, they look healthy, but they don't have an acrosome. This is another genetic anomaly that does not allow a male to make an embryo from his own sperm. So although morphologically complete, spermatozoa released from the seminiferous epithelium into the lumen of the tubule, they're still functionally immature because they need another 10 to 12 days where they undergo additional biochemical modification. These changes allow the sperm to acquire progressive motility and the ability to undergo capacitation in the female reproductive tract. Here we have surface proteins such as CRISP1, cysteine-rich secretory protein 1, which plays a role in zona pellucida binding, and P34H, which is involved in sperm egg recognition. Again, very important. Without these markers, it is still going to be, this sperm will still be deemed as functionally immature and impossible to create an embryo. So now the breakthrough, the innovative approach in the management of absolute male infertility. Like I mentioned in the process of spermatogenesis, what can go wrong in males where inside the spermatogenesis itself, where the sperm can be present, but still be functionally absolutely immature or structurally immature, or sperm not being present at all. Also, chromosomal and genetical disorders can have that. So this is a patented content alert. I would like to tell you again that this is a patented technology. So it cannot be used in any other lab other than the Ay.D stem cell and genome lab.
We have registered this patent already with 143 countries. And just with a few months ago, that have full rights in Malaysia, in Indonesia, Malaysia. In Malaysia, one university, Malaysian university, they have a full right to do this technical in their lab, but there is some analog technology that they are doing in Japan, in China, and in Brazil. But our technology, what we are doing, there is nothing from outside from the body, not using. We didn't use anything from the another biologic donors or only from the same body. We didn't use anything which chemical or donors. There is some technique which we are block some gene which blocked the spermatogenesis in the one from why Dr. Aminish spoke with you that there is different step of the spermatogenesis because we have some technical that we can block this atypical gene which we have in our body. Okay, you can continue.
Okay, so now I'll speak more about the technique itself. So one of the main and the initial stages of the technique of spermatogenesis out of the patient's blood is identifying the stem cells that can be further differentiated into any type of germ cell or spermatocell or oocyte. So at first, our patients receive GCSF factor. They receive it to stimulate the bone marrow, which helps it mobilize the stem cells into the peripheral blood. After 24 hours, we collect approximately 300 to 400 milliliters of blood because to find those extremely pluripotent stem cells, we need a little too much blood because they are rare, but they are present. But they're rare. So you need enough blood to find them inside of it. So after that, we filter it, we isolate the MSCs, obviously with the presence of heparin and the 100 ml, which is added for anticoagulation. Key markers of identifying mesenchymal stem cells in any type of tissue, from adult, from the adipose tissue, from blood, from umbilical cord blood, even the placenta. The markers for identifying mesenchymal stem cells are the same all throughout, which is CD105, CD73, and CD90. These are the standard MSC markers. So this is something you need to remember at all times for future studies about the regenerative medicine. After microscopic examination, approximately 95 to 98% viability in polymorphic nucleus-containing cells of various sizes, which means that with the identifying markers, we will have approximately 95 to 98% of the cells present in the withdrawn blood as MSCs. After that, they are concentrated into 2 milliliter portions by centrifugation. So these cells are given as IV as well for the patient, because then we stimulate his body to regenerate, we stimulate his body to have some new processes. So when we give him, it's basically like anti-aging process. The same technique is for the isolation of the sperm. Again, we identify MSCs from the withdrawn blood. They are marked with CD105, 105, 73, and 90. After that, they are additionally stained for CD133, which is a marker that is commonly associated with an embryonic origin and pluripotency. This aids in identifying stem cells with higher regenerative potential. So this analysis in our lab revealed that approximately 15 to 20% of the MSCs, which means the stem cells that are stained positively for 3 markers, 105, 73, and 90, approximately 15 to 20% are also stained for the CD133, which means that not all mesenchymal stem cells derived from your own bone marrow are pluripotent. Approximately 15 to 20% of those cells are pluripotent and have an extremely high potential for further proliferation. So this is an image of the MSC pool that was obtained. So you can identify here, there is erythrocytes and there is stem cells. Stem cells, they usually come in clumps. They come together. They stick together all the time. These are stem cells. These are stem cells. They always like to be together, which creates it easier to filtrate them and makes it easier for them to communicate and stay pluripotent as they are. Also, like, you can identify them by the presence of small nanovesicles called exosomes. Maybe the next lecture we will speak about exosomes. Step 2, to further refine this continuation. So to further refine identification, the isolated MSCs are also tested for OCT4, octamer 4. This is the key marker of pluripotency. Just so you know, anything from I.D. Stem Cell and Genome Institute, if it's not tested positive for or marked positive with OCT4, we do not use it, because this is the best marker of our body, of the human body, that is present for pluripotency and undifferentiated state. So approximately 75% of the MSCs that were positively stained with all the MSC markers, and then the pluripotency CD133 marker, 75% of those stained positive for OCT4 as well. This means that CD133 and OCT4 are the main markers for identifying pluripotency, which is what we're looking for, because the pluripotency, it is also very popular among some scientists to believe that pluripotent cells are not present in our body. This is what Professor Islam and his team are strongly advising against, because we do have pluripotent cells inside of our body. Yes, they're in very little amounts, and the older you grow, the lower is the count of those cells, but they are present, because they're what helps our body stay alive in case of emergencies. So these properties indicate the possibility of the cells to be germ cells, which is the pluripotent cell that can further develop into any sexual type of cell, which means that they are present in the human body. After that, the isolated and identified cells, they are cultured in a special designed environment that mimics embryonal germ cell conditions to promote their differentiation into spermatocytes. So the goal here is to create an environment for those pluripotent stem cells so that they get the signal that they're supposed to become the spermatogonial stem cells, because this is how the stem cell doesn't die. It either goes to sleep, dies, or becomes a differentiated cell. So to help it differentiate into something, you need to give it the proper signal. Inside the human body, it takes whatever signal it gets. So whether it is a local injection somewhere, the stem cell gets the signal from the cells around it and then understand what it needs to become. Here in vitro, we need to give it the signal that it needs to hear, that we want it to hear. Another method of induced pluripotency, but without the genetic modification. By day 13, we observed that these cells started expressing DAZL, which is a marker that is essential for the initiation of meiosis. Not meiosis of spermatocytes, but meiosis in general that will help to go from diploid to haploid state of the cell. Again, this is the MSC pool that is stained for OCT4, the brown dots. These are the cells that are stained for OCT4. Just like we mentioned, 15 to 20% are positive for CD133, and out of these stained for CD133, they're also stained with the OCT4, which are in brown color. But this is still in the pool derived from the blood. So these are all mesenchymal stem cells, but only this little amount of them are actually genuinely pluripotent. These are the ones that stained positive with DAZL. You can see that they have this granule golden brown color, which is the hallmark of DAZL. By day 15, the majority of cells express DMC1, which is a marker of homologous recombination during the meiosis. So this confirms that the meiosis is successful. After that, it is supported by the theory that both the reproductive and hematopoietic systems originate from the mesoderm, which means that from your blood, we can grow sperm, specifically from the stem cells that are taken out from the hematopoietic system, because they have the same origin in the embryo, the mesoderm. Starting on day 32, the sperm starts developing acrosine, which is a protease enzyme, like I explained before, crucial for spermatogenesis, begins to form at the periphery of the cells. By day 32 to 45, acrosine granules concentrate at one pole of the cell, usually at the right above it, and by day 45, the spermatocyte display motility, indicating their maturation. So we want to say that by day 45 in vitro, the sperm is already fully developed.
Here, very important, we want to show that we added something in the media, what we are using. Not only that the cells, it's growing by themselves. No, we added some testosterone, some another materials or some active materials we added to the media in the stem cells media.
Yeah, so like I mentioned, the media is what helps it get the signal to grow into whatever we want the cell to grow into. But obviously the media cannot be performed once, cannot be done once. It needs constant
Every 3 days, every 3 days, we add in some new active substance.
Yes.
Yeah.
Okay, here are the most interesting parts. So the DMC1 is the marker of successful meiosis, like we mentioned. These are the stained cells with DMC1. And here, as you can see in the cells, we have acrosine, brown granules at the poles of the cell, like around the cell. But in some cell later, we can see that the majority of it are accumulating at only one side, like here, they're accumulating at one side at some time. So within this time period from day 32 to 45, they start accumulating from around the cell into one pole only. After that, obviously, we can take this sperm. So I'll tell you exactly what happened in our study. So we took some part of the sperm cells on day 45 because they exhibited full structural and morphological maturity. We took them and attempted in vitro fertilization. And it was approximately successful, 20 to 35%. However, till we had to give them the chance till day 75, like the full cycle of spermatogenesis inside the human body. What we noticed is that from day 45 to 75, the main morphological structural thing that was changing is just the tail. The tail was growing, it was becoming more mobile, but there is also a negative side of it. The longer it is, the more mobile it is, the harder it is to catch it, and the easier it is to break later before the in vitro fertilization. These are mature gametes. I think we also have a video for it. If we will be able to show you. Oh, yes, this is the video. So this is the oocyte. So all these cells that you can see in the media, they have the structural, morphological structure of sperm. They have the main body, they have the tail, they have, in some of them you can notice the acrosine, and they're all going around the oocyte. Please tell me you were able to see the video. Hello?
Jesus, what culture?
Okay, so you were able to see. Great, amazing. Okay, so we will go to the next slide. So this is where we start after completing the study. This is where we start identifying safety and future research and clinical application need. Obviously, it needs a more extensive clinical trial. It needs a more extensive stages to identify all possible. What's important is the quantity of the patients, because we cannot say a certain statistics at this stage where any faults can start growing or not with babies that are done from such a technique, from such a sperm developed fully in vitro. This obviously needs. We have a few patients, but we cannot tell you the full statistics from huge clinical trials. It obviously needs years of
less than 100 patients, which was did, but still it's not enough to talk about this mechanism of treatment or these protocols as a standard for the treatment. Only in Malaysia they take it and they recommended the MOH that this technique can be using as one of the protocol of the treatment, and here in Ukraine we're using already as one of the protocols. But still we need more patients, still we need more specialists who can do with us this trials.
Special thanks to our partners in all of the universities that we partner with in Ukraine and across the world, the US and Europe and the UAE. So thank you. This is it.
We have around 10 minutes. If anyone have any question want to ask, we can answer you. We have 10 minutes for this.
So in which country we are based? We are based in Kyiv, Ukraine. Our main lab and our main clinic and hospital are here. Our main team is here as well. When can we apply this study on patients? We are already applying it for patients.
Yes, hello, doctor. Can I open the mic?
Yes.
First, thank you for this nice presentation. I'm actually interested in the stem cell field. I am a biomedical lab technologist and ISO accreditation also, and CAP accredited, and AABB accreditation specialist. My concern here, I ask you the first question, in which country you are based, because I saw the symptom of Abu Dhabi. So I thought that you have even a branch here in Abu Dhabi.
We have a good relation with them. We are doing something with them.
They are our partners.
They are partners. Okay. I have another concern also. Is there any side effect for the stem cell in this process? Like the genetic, like any other side effect?
Before then we do embryo transfer, we doing the PGT for all our baby or embryo what we have, and we investigate them in 2 different labs.
So you will make like comparison for the reports.
Exactly.
Before you start the process.
Exactly.
Yeah, this is number one question. The second question, I think the process you are doing is nearly to the IVF process.
It's one part from them, from the IVF.
Yeah, the different, you said this is all the processes inside the body you will do. And but for the IVF, there is another technique that they are doing external, especially
we do everything external. We just the first day we stimulate the bone marrow, and we taking the blood, and we making everything outside.
Outside, and everything is perfect, then you will inject it inside the body.
Exactly.
I thought the technique will be like the IVF, because the IVF they do it outside. Maybe they do incubation, and then they
Everything is here is the same, that we are taking the cells, we have some manipulation with the cells, we blocked the gene, what we have problem with them, we growth another new gene with another technical, and we grow the spermatogenesis. The first thing we need to find the gamete. This is very important. From germ cell to the germ cell to gamete. This is our goal. If we cannot transfer the germ cell to genome, we will not go to the next step. Here, I want that we have two ways to treat the patient. Sometimes we are doing everything in the lab, and we growth from day 30. We growth the sperm in the lab. The second technique, we can inject this sperm in the first stage in the testes sometimes, and after 53 days we can make taser and take this still and do the ingester.
But then again, this
I
is
go.
extremely personal to patient, and depends on the patient's choice. For the males, it's usually an extremely painful procedure, so they choose just to proceed with the with the in vitro.
Second in vitro.
Yeah.
Thank you so much.
Thank you as well. So we have another question. If you have any ideas about the future of IVG, in vitro gametogenesis, based on using stem cells. This is basically what we're talking about. This is what the webinar.
This is what we are doing. Exactly.
Yeah, so this is in vitro gametogenesis. Hopefully in the future, Professor Islam gets another bright idea of how to develop an oocyte in vitro. And this is kind of our main goal as well for women that have.
We have a protocol. We have a protocol exactly that to do this, but we have, I think, 2 patients only. We did, and we still not successful with this. Any trials done in UAE? In UAE, very difficult to do any trials. I try with already 3 years. From Egypt, that it can be.
Can you hear us?
Hello?
Hello, doctor.
Yeah. Okay.
Doctor, I asked you if can we apply the situation on patients?
Yes.
Exactly. Regarding this, the patient, we will check the patient, we will investigate with 14 days, we will tell the patient which kind of the treatment we will do. Sometimes we just inject it in the testis stem cell, and this is enough if there is no genetic problems. And this can be enough to stimulate. We have a small book for this. We did with 20 page. I think our assistant, they can send for you this box. It's just simple box, not scientist box. It's just only for the simple thing to do for the people, that what we are doing exactly.
Doctor, my husband have genetic problem, clean filter.
If you have a clean filter, or husband have a clean filter, we need to do nick out of the gene. Gene nick out. That maybe to talk for you in Arabic better for you, or to keep in English. يعني you have X, Y, right?
Right, doctor.
We need to block this one X. Okay?
Yeah.
We need to block. What's here? You see me, right?
Yeah, I see, doctor.
This is the locus. This is the locus of the game. If you know this, I don't know if I have any picture here to show you. It's like this, okay? And here we have a locus.
Doctor, we make him a sperm inspection, and we take a microscopic histology. The result, no spermatid and no spermatozoa. Most of the tubules are populated by sterile cells only. Only few tubules contain germ cells, spermatozoa, and spermatids.
You need to growth in the lab. They need to take and to grow this in the lab.
Can we? Possible?
It's possible, exactly.
Yeah, maybe you would like to get a personal consultation with us.
We can
Yeah.
growth, and there is many. There is electrostimulation for the sperm in the world. Not only we are doing, it's a Japanian patent. They are doing, and it's working. They can growth.
I hope.
Yeah, there is many. If you have spermatogenesis and you have first stage of the sperm, they can growth in the lab. Not only in our lab.
Doctor.
Yeah.
But they told us there is no hope.
Yeah, because in the books, a genetic disorder that gives you zero sperm cell means that you cannot produce an embryo yourself. Physiologically, it is impossible. That doesn't mean that new techniques in regenerative medicine are not going to make this possible. This was the whole point of this webinar. We understand that in the MENA region, it is somewhat very difficult for the people ethically to dive deep into this science, but this is science. This is somewhat like 100 years ago, augmentin was absolutely impossible to believe in. And antibiotics were absolutely impossible to believe in. Something that will help kill the bacteria, as simple as what you can go and get from the pharmacy easily right now. 100 years ago, maybe 50 years ago, they were absolutely impossible to believe in, and the people did not understand it.
And I am sorry that in Middle East and MENA, the IVF center, they start only in 1996. But in general, in the world, 1972. يعني we have a
1972. So we have approximately like a 30-year
25
delay.
years delayed.
Yeah, delay in the MENA region of
Okay.
all the science. This is all trying to make it
there is someone asking the percentage of sexual and can use for the female and male, how many time we need for this. The first thing, if you're asking in Arabic, maybe you don't know very well English, I will answer in the Arabic and translate to English. Okay. كم نسبة نجاح العملية، وهل تنفع للزوج والزوجة، وكم تستغرق من الوقت؟ نسبة النجاح, we still doing this trials. We need more in Arabic. I will go back. لسه احنا في عملية الأبحاث السريرية، نحتاج عدد مرضى أكثر وأكثر. نسبة النجاح إلى اليوم احنا تتراوح ما بين 35 لل 40%، مع تحسن بين فترة وفترة، لأنه بيختلف من جسم إلى جسم، وإنه المجسم أو الرسمة اللي بنعملها الجينية تختلف من مريض إلى آخر. فلذلك هي تحتاج إلى أكثر عدد مرضى حتى نستطيع إنه يكون هذا بروتوكول دولي وعالمي وشغال. إنه نقدر إحنا نعطي نصائح للمريض إنه أنت لازم تروح بهاي الطريقة. إلى الآن إحنا في الأبحاث السريرية في هذا الموضوع، ونرجو من الله أنه يتم استقبال هذا الحكي في العديد من الدول العربية. في هناك بعض الدول العربية ومنها يعني الإمارات العربية المتحدة، الحمد لله أنه إحنا أكثر من مرة كان لنا محاضرات فيها. ماليزيا، إسبانيا، وين كمان، إيطاليا، فرنسا، ألمانيا. يعني كان في عندنا لكتشرات، دروس في هذا. تعمل للمرأة والرجل؟ نعم، تعمل للمرأة والرجل. كم تستغرق؟ للمرأة تقريبا تحتاج تقريبا شهر in the lab. للرجل نحتاج تقريبا من 3 شهور إلى 6 شهور. بجاوب بالإنجليزي بنسبة يعني سريعة، بطريقة سريعة. Our percentage of successful is still 35 till 40%. It's can be done for the female and male. Yes, can be done. How many time we need for the female? We need 1 month. For the man, we need from 3 months till 6 months. Next one.
Is there
it's-
any dates for publishing the results?
E-
So the final results, I'll explain a little more. We have done this study. We finished the study in 2022. We published it in Ukraine in 2022. After that, we sent it for a patent. To explain what a PCT patent is when you give your technique to an independent committee to test it. So a team across 11 different universities across the world, which we did not know about, we still don't know what universities and what people did
We
the
don't
study,
know them.
that we don't know them, have no contact with. Independently, 11 different teams did the study according to the technique that we provided. Our scientists did this, the technique we provided to them, they did it themselves absolutely. After that, after 3 years, they have finally, even 3 and a few months, they finally proved that this technique is possible, is functional, and can be used. So the results are, we can't say exactly because the statistics, every patient has their own diagnosis, has their own problems, has their own mix of genetic and chromosomal abnormalities, as well as environmental and morphological specialties. So every case is independent. A certain statistics on this specific method cannot be told or calculated, but in general, from the patients that we have at I.D. clinic, we would say 30 to 40% are successful.
No, go back here.
Okay.
We have some publication. We did one publication or many publication. We have a many lecture in international conference. Can we advise to
Any country to make this? We're doing this in Ukraine.
We're doing this in Ukraine. You can go to Japan, China.
Not with us.
Not
Japan
with
and
us.
China do it by themselves.
By
If you
themselves.
want the technique that we do it only in Ukraine.
Only in Ukraine. احنا بنقوم بهاي العملية، في سؤال بالعربي، بنقوم في هاي العمليات في أوكرانيا حاليا. كم عملية العمليات؟ لما أقول أنا في عندي 35% ل 40% نسبة نجاح، يعني أنا عملت تقريبا 100 حالة، منهن 35 حالة نجحت. ممكن التعاون نعمل في سوريا. احنا بنتشرف أكيد في أي مختبر أو أي كلينيك حابة نعمل في هذا المجال معهم، وإنه يتم هذا بشكل قانوني، بشكل حسب متطلبات وزارة الصحة وفي الدولة اللي بنكون بدنا نتعاون معها. ممكن عمل هذا التعاون عن طريق الدولة كمان. يعني احنا الحين في صدد توقيع اتفاقية في أحد الدول عن طريق إرسال الساينتيست تبعينا إلى أحد الدول العربية. كيف يمكن التواصل مع المختبر؟ أكيد تواصلوا عن طريق كيف سجلتوا عندنا على الويبينار. ممكن تتواصلوا معنا بنفس الطريقة علشان تحصلوا على أكثر معلومات. بالنسبة في حال فشلت العملية وتكاليف عالية، هل يمكن الإعادة بضمان تخفيف أو الإعادة على حسابكم؟ طيب، أول شغلة إحنا كل ستيج بالنسبة للعلاج بناخذ مبلغ لحاله. يعني علشان نلاقي جيرم سيل هذا بناخذ مبلغ لحاله، علشان نحول الجيرم سيل لا جيميت بناخذ مبلغ لحاله. فكل مرحلة بمرحلتها. يعني احنا ما بناخذ كل المبلغ كله مرة واحدة. كويس. وبعدين بنقول لك والله احنا بعد شهرين والله ما قدرنا نحصل على حاجة. لا، احنا اذا حصلنا على المرحلة الاولى ناخذ اول مبلغ علشان نسوي المرحلة الاولى، اذا ما حصلنا ما بناخذ مبلغ المرحلة الثانية. مساعدة شو اسمه. أكيد إحنا دائما بننظر بالإمكانيات اللي بنقدر عليها. وفي عندنا حالات ساعدناهم ما قدروش يكملوا بالمرحلة، يعني ما نجحت معهم ما قدروا يكملوا، وعملنا على مساعدتهم. وهذا بالطبع مش ضروري أحكي كم حالة أو كم شو اسمه، هذا يتم عمله لوجه الله. Anything else؟ في أي حدا عنده أي سؤال؟
It's not a question. هو ما سؤال يا دكتور، بس ربنا يوفقكم إن شاء الله. وده برنامج يعني هتدوا فيه هوب كثير لكثير من الناس. الحاجة اللي عايز أسأل منها هو سؤال عابر. هل أنت بتحتاج لأنك تاخذ أبروف من ال WHO بالنسبة للبروجكت هذا ولا ما تحتاج؟
لا، احنا اللي هي في عندنا دبليو اتش او ما بدنا منهم، لانه البروتوكولز بيتم اعتراف فيه حسب الدوله. احنا يا اما بنروح على الاف دي اي ابروفال. الاف دي اي لحد الان مانع موضوع الخلايا الجذعيه، لانه اتس وركينج اجينست، شغاله ضد شركات الادويه. يعني انت لما تقول انه انا في عندي حل مثلا لمرض السكري، وهم عندهم ميزانيه، الاف دي اي عندهم ميزانيه 5% من البادجت الامريكي على بيع الادويه لمرضى السكر. انت قاعد يعني بتحارب في شركات الادويه. كم تبلغ
شغال
التكاليف؟ آه
شغال معانا بالعكس يعني.
ايه، شغال معنا بالعكس اكيد.
مم.
في حال كم تبلغ التكاليف؟ بالطبع التكاليف تختلف من حالة لحالة، وشو نوع العلاج اللي راح يتم استخدامه. يعني شو العلاج اللي احنا راح نقوم فيه؟ هل هو بس حقن الخلايا الجذعية؟ هل هو نعمل موديفيكيشن على الخلايا الجذعية؟ هل احنا بدنا نعمل شو اللي بدنا نعمله في الخلية؟ هل بدنا نعمل نيك اوت بلوك لجين؟ هل بدنا نعمل؟ وحاليا صراحة كل يوم الأسعار بتختلف حسب إمكانياتنا وقدراتنا إنه نحصل على المواد الأولية. هل في تجهيز قبل العملية، أي أدوية أو أي بروتوكول معين؟ نعم. إذا حضرت المحاضرة بالكامل في تجهيز قبل ما نأخذ العينة من الرجل، في تجهيز بنعمله. نعم صحيح.
وما بعده في فيتامينات
حسب
و
نرجع
it's
ونقول
personal.
حسب الحالة اللي بدنا. في حالات ما بنعمل تجهيز، في حالات ما بنعمل حقن، كافي ناخذ عينة وممكن نلاقي لنا 1 سيلز جيرم، يعني ناخذ 1500 15 مليلتر وكافيتنا نلاقي 1 سيلز، واتس انف انه نعمل لها استنساخ. نعم، حسب الحالة، حسب منين بناخذ الدم، حسب وين بناخذ الدم، يعني في امور كثير بتترتب على هذا. في بعض الأحيان بالطبع في دول عربية في إمكانية الوصول لأوكرانيا حاليا للعلاج. بنقدر نعمل دعوات ونعمل فيزا ونستقبل، وعندنا حالات بيجوا من دول مختلفة.
If you're asking about how can you contact us, I have sent our number that you can contact through WhatsApp or Viber. You can contact us through Instagram and the email that I have sent. We tried our best to respond as fast as possible.
وكذلك في عندنا زي ما دخلتوا، إذا بتقعدوا تدخلوا على الموقع، في الموقع أمور كثيرة مشروحة، وفي طرق كيف التواصل، وفي فريق كامل متكامل لمساعدتكم للوصول للاستشارات الطبية. شكرا للفريق السبورتينج تيم، يعطيكم العافية. شكرا.